Hello and welcome to the channel. Tonight we're exploring a question that echoes through hospital hallways, research labs and quiet moments of fear. Why is cancer so hard to kill? It moves like a whisper beneath the surface. Not foreign, but part of us, cells that forget the rules, that choose chaos over harmony, multiplying in silence until they change everything. There is mystery here, not just in the science but in the heartbreak. How something born from within can turn against us. How brilliant minds race against time, and yet the answers still slip just out of reach. And yes, it's curious. This powerful, complex topic, meant to ease you into sleep. But sometimes, understanding the quiet war within us can bring peace. Sometimes the mind softens when it finally feels seen. If this gentle corner of knowledge brings you comfort, please consider liking or subscribing. It helps keep the light on here for you and for others who find rest in wonder. And if you're still with me, leave a comment below. Tell us where you're listening from, or what time it is right now. Our little nighttime community is scattered beautifully across the world, connected by curiosity and the need to breathe a little easier in the dark. So now, breathe in deeply, and let it go. Soften your shoulders, unclench your jaw, let your body melt into stillness, like snow disappearing into the ocean. You're safe here. You're not alone. And as your breath slows, we'll gently begin to unfold the story of cancer, why it hides so well, fights so hard, and why, despite everything, we are learning, slowly, beautifully, how to fight back. What cancer really is. A rebellion of cells. Picture your body as a vast, bustling city. Not New York or Tokyo, but something more like a perfectly orchestrated metropolis that never sleeps 24 hours a day, 7 days a week. Construction crews are building, repair teams are fixing, and waste management is cleaning up. Everyone has a job, everyone follows the rules, and somehow, miraculously, it all works. Your skin cells know exactly how fast to replace themselves. Not too quickly, or you'd shed like a snake. Not too slowly, or cuts would never heal. Your liver cells regenerate with mathematical precision. Your heart cells beat in rhythm for decades without missing their cue. It's a symphony of cooperation that would make the most advanced AI weep with envy. But then, something goes wrong. Imagine if one construction crew in this perfect city suddenly decided they didn't need permits anymore. They didn't need to follow building codes. They didn't need to stop when the project was finished. They just kept building, higher and higher, using more and more resources, ignoring every stop signal the city sent them. That's cancer. Not some alien invader. Not some mysterious disease from outer space. Cancer is what happens when our own cells that were once good citizens in the body's cooperative society, become rebels. But here's what makes this rebellion so insidious. These rebel cells don't look completely foreign to the body's security system. They're not wearing black hats and carrying signs that say "I'm the bad guy." They're your cells, with your DNA, speaking your body's language. They've just changed the rules they live by. Bert Vogelstein, one of cancer's most persistent hunters, once described cancer as a disease of too much life, not too little. While we often think of disease as things shutting down, cancer is the opposite. It's cells that have forgotten how to stop living, how to die when they're supposed to, how to stay in their designated neighborhoods. Every day, your body produces millions of new cells, and every day, some of those cells make mistakes. Tiny copying errors in their genetic code, like typos in an incredibly long document. Most of the time, your body catches these errors. It has proofreaders, spell checkers, and quality control inspectors working around the clock. When they find a cell that's made too many mistakes, they calmly escort it to cell death, a process called apoptosis, which sounds harsh but is actually as natural as autumn leaves falling from trees. But sometimes, just sometimes, a cell accumulates the right combination of mistakes. Mistakes that make it ignore the stop signals. Mistakes that make it demand more food than its fair share. Mistakes that teach it to hide from the body's security forces. These aren't random mistakes. They're like a criminal learning exactly which laws to break, to avoid detection. The cell begins to divide when it shouldn't. It moves to places it doesn't belong. It sends out chemical signals that trick other cells into helping it. It's not just rebelling, it's recruiting, like a charismatic leader starting a revolution. And here's the truly unsettling part. This rebel cell isn't trying to destroy the body. It doesn't have consciousness or malice. It's simply following its new corrupted programming with the same dedication that your heart cells follow theirs. It's doing what it now believes it's supposed to do, which is grow, spread, and survive at any cost. This is why cancer is so different from fighting bacteria or viruses. When you take an antibiotic, you're trying to kill something that's genuinely foreign to your body. But fighting cancer means fighting yourself, or rather, fighting the version of yourself that's forgotten how to play by the rules. Imagine trying to stop a revolution where the revolutionaries look exactly like loyal citizens, speak the same language, and know all the secret passwords. Imagine trying to restore order to a city where the construction crews have all the right permits. They just happen to be forged. That's the fundamental challenge of cancer treatment. We're not trying to kill an invader, we're trying to convince rebels to stop rebelling, or eliminate them without destroying the loyal citizens who happen to live right next door. And that brings us to perhaps the most important thing to understand about cancer. It's not one disease. When we say cancer, we're really talking about hundreds of different rebellions, each with its own personality, its own tactics, its own weaknesses. Lung cancer behaves differently than breast cancer. Even two people with the same type of breast cancer might have tumors that are as different as snowflakes. This is why your grandmother might have beaten breast cancer in 6 months, while her friend with the same diagnosis had a completely different journey. The rebels in their bodies learned different tricks, chose different strategies, responded to different negotiations. Some cancers are like organized crime, methodical, strategic, patient. Others are like street riots, chaotic, fast-moving, destructive. Some are like master spies, hiding so well that the body's immune system walks right past them. Others are like bank robbers, so obvious and aggressive that every alarm in the body starts screaming. Understanding this diversity is crucial, because it explains why there's no single silver bullet for cancer. There's no one cure, because there's no one disease. There are hundreds of different rebellions, each requiring its own strategy, its own negotiation tactics, its own form of peacekeeping. But before we despair at this complexity, remember, every revolution in history has eventually been understood, predicted, and managed. The human body is incredibly resilient, incredibly clever, and incredibly determined to maintain order. We're learning to speak the language of these cellular rebels, to understand their motivations, to predict their next moves. The war is far from over, but we're no longer fighting blind. We're beginning to understand our enemy, and our enemy, as it turns out, is also ourselves. Mutation Nation. How cancer evolves from within. There's something almost Darwinian happening inside every tumor, something that would fascinate Charles Darwin himself if he could peer through a microscope into the cellular world, because cancer doesn't just grow, it evolves, adapts and learns, like a species fighting for survival in the hostile environment of your body. Imagine a city where every building is constantly under renovation, but the construction crews keep making small changes to the blueprints. Most changes make the building worse. A window in the wrong place, a door that opens onto empty air, a staircase that leads nowhere. But occasionally, purely by accident, one of these random changes makes the building stronger, more efficient, or better able to withstand storms. Now imagine this process happening millions of times, in millions of buildings, over months or years. The buildings that survive are the ones that accidentally discovered better ways to exist, the ones that didn't simply disappear. This is evolution in fast forward, and it's happening inside tumors every single day. When a normal cell first becomes cancerous, it's actually quite fragile. Like a freshman politician who's just learned to bend the rules, it's making lots of mistakes, attracting attention, and generally struggling to survive. Most of these early cancer cells don't make it. Your immune system spots them, your body's quality control systems eliminate them, or they simply can't figure out how to get the resources they need. But the ones that do survive, they're the winners of a ruthless genetic lottery. Dr. Peter Nowell first described this process in 1976, calling it clonal evolution. He realized that tumors aren't static lumps of identical bad cells, they're more like ecosystems, filled with different populations of cancer cells, each carrying slightly different genetic mistakes, each trying different survival strategies. Think of it like this. If the original cancer cell is like a master criminal, the tumor becomes a criminal organization, where each member has learned different specialties. Some cells become experts at avoiding the immune system, others excel at demanding more blood supply, still others master the art of breaking down barriers and moving to new territories. But here's where it gets really unsettling. This evolution never stops. Even while doctors are treating the cancer, even while chemotherapy is killing billions of cancer cells, the process of mutation and selection continues. It's like trying to eliminate crime in a city where criminals are constantly learning new techniques, adapting to every law enforcement strategy you deploy. Cancer cells make mistakes when they copy their DNA at a rate that would be catastrophic for normal cells. A healthy cell might make one mistake per million letters of genetic code. Cancer cells can make hundreds or thousands of mistakes. Normally this sloppiness would be fatal, like a construction crew that can't read blueprints properly. But cancer cells have also broken the quality control systems that would normally catch these errors. So instead of dying from their mistakes, they live with them. And occasionally, one of these mistakes turns out to be an advantage. Maybe a mutation helps the cell ignore a stop-growing signal from neighboring cells. Maybe another mutation helps it hide from immune system patrols. Maybe a third mutation teaches it to survive in low-oxygen environments, like the center of a growing tumor where blood supply is limited. Each advantageous mutation spreads through the tumor like a useful innovation spreading through a company. The cells with better survival skills outcompete their neighbors, have more offspring, and gradually dominate the tumor population. This is why cancer can become resistant to treatments that initially worked well. Imagine treating a tumor with chemotherapy. The drug kills 99.9% of the cancer cells, a resounding success by most measures, but that remaining 0.1% weren't killed because they had some genetic quirk that made them resistant to the treatment. Maybe they had better DNA repair systems, or more efficient drug pumps that could expel the chemotherapy before it did damage. Now these resistant cells have the entire tumor to themselves. They multiply, carrying their resistance with them, and when the cancer returns, it's populated entirely by cells that are immune to the treatment that worked so well the first time. It's evolution under pressure, and the pressure is our own attempts to cure the disease. Dr. Mel Greaves, a British cancer researcher, uses a particularly striking analogy. He says treating cancer with a single drug is like trying to eliminate weeds from a garden using only one type of herbicide. Initially, most weeds die, but the few that survive will be the ones with natural resistance to that herbicide. Soon your garden is overrun with superweeds that laugh at your best efforts. This is why modern cancer treatment increasingly uses combination therapies, multiple drugs that attack different aspects of cancer cell survival. The idea is to make it nearly impossible for any single cell to have resistance to all the treatments simultaneously. It's like trying to survive in a city where you need to simultaneously avoid the police, the FBI, the tax authorities, and the building inspectors. Few criminals are that versatile. But even combination therapies face the challenge of cancer's evolutionary creativity. Tumors can develop resistance through pathways that scientists never anticipated. They're like master escape artists, who always seem to find the one weakness in even the most secure prison. Some cancers learn to repair DNA damage more efficiently, essentially developing better healing abilities. Others master the art of going dormant, entering a kind of cellular hibernation where they're nearly invisible to both treatments and the immune system, only to wake up months or years later when the coast is clear. Perhaps most remarkably, some tumors develop what scientists call phenotypic plasticity, the ability to change their fundamental identity when under threat. It's like a bank robber who can instantly transform into a police officer when the real police arrive. Certain cancer cells can temporarily stop behaving like cancer cells, mimicking normal cells so convincingly that treatments designed to target cancer leave them complete alone. This evolutionary arms race helps explain why cancer research can feel like an endless game of whack-a-mole. Every time scientists develop a new weapon against cancer, evolution finds a way around it. But this isn't a reason for despair, it's a reason for smarter strategy. Understanding cancer as an evolutionary process has revolutionized how researchers approach treatment. Instead of just trying to kill cancer cells, they're learning to guide cancer evolution, to steer it toward less dangerous paths, or to create evolutionary traps where cancer's own adaptability becomes its weakness. Some researchers are exploring adaptive therapy, deliberately giving cancer cells more genetic diversity, then exploiting that diversity with rapidly changing treatment combinations. Others are working on evolutionary steering, where treatments are designed not just to kill cancer, but to force it to evolve in directions that make it more vulnerable to future attacks. It's a chess game played at the molecular level, where understanding your opponent's strategy is just as important as having powerful pieces. And cancer, it turns out is a remarkably sophisticated opponent, one that's been perfecting its game for as long as multicellular life has existed. But remember, evolution also gives us hope, because if cancer cells can evolve, that means they can also be outsmarted, outmaneuvered, and eventually defeated by opponents who understand the rules of the evolutionary game better than they do. The body's betrayal. When healing becomes harmful, there's a cruel irony at the heart of cancer that keeps oncologists awake at night. Many of the processes that cancer exploits, the very mechanisms that make it so dangerous, are actually our body's most generous, life-giving systems. Cancer doesn't just hijack our cells, it corrupts our body's capacity for healing, turning our greatest biological gifts into weapons against us. Imagine if a con artist didn't just steal your money, but somehow convinced your own charity work to fund their criminal enterprise. That's essentially what cancer does with the body's healing systems. Let's start with something beautiful: angiogenesis, the process by which your body grows new blood vessels. When you cut your finger, within hours, your body begins the delicate work of weaving new capillaries to bring nutrients and oxygen to the healing tissue. It's like your body is knitting itself back together with threads of life. This process is regulated with exquisite precision. Your body produces signals that say grow blood vessels here, and other signals that say stop, we have enough. It's a conversation between your tissues, a molecular negotiation that usually results in exactly the right amount of blood supply. No more, no less. But cancer cells are master manipulators of this conversation. Dr. Judah Folkman, who pioneered our understanding of tumor angiogenesis, discovered that cancer cells can essentially hijack your body's blood vessel construction crew. They send out distress signals that mimic the chemical cries for help that injured tissue produces. "We need blood vessels," they scream. "We're starving over here." Your body, being the generous landlord it is, responds by growing an elaborate network of blood vessels into the tumor. But these aren't the neat, organized blood vessels that supply healthy tissue. They're chaotic, leaky, poorly constructed highways that look more like the work of a corrupt construction company than your body's usual precision engineering. Cancer doesn't just trick your body into feeding it. It turns your body's healing response into a tumor growth accelerator. The same thing happens with inflammation, another of your body's most sophisticated repair systems. When you sprain your ankle, the swelling, heat, and redness you experience aren't signs that something's going wrong. They're signs that your body's emergency response team has arrived. Inflammatory cells rush to the scene, cleaning up damaged tissue, fighting potential infections, and coordinating the repair process. But chronic inflammation around tumors tells a different story. Cancer cells produce signals that keep inflammatory cells perpetually activated, like keeping the fire department permanently stationed in your neighborhood. These inflammatory cells, thinking they're helping heal injured tissue, actually end up providing growth factors that feed the tumor, breaking down barriers that would normally contain it, and even suppressing other immune cells that might recognize the cancer as a threat. It's like hiring security guards who end up working for the burglars. Perhaps most heartbreaking is how cancer corrupts the immune system's capacity for tolerance. Your immune system is naturally programmed not to attack healthy tissue. It has to learn the difference between self and foreign, between friend and foe. This tolerance is essential. Without it, you'd have autoimmune diseases where your body constantly attacks itself. Cancer exploits this tolerance with sophisticated deception. Many tumors learn to display molecular signals that say I'm supposed to be here or I'm healthy tissue that shouldn't be attacked. They waive the biological equivalent of diplomatic immunity, convincing immune cells to leave them alone. Some cancers even recruit regulatory immune cells, the very cells whose job it is to prevent excessive immune responses, and use them as bodyguards. These regulatory cells create a protective shield around the tumor, actively suppressing any immune cells that might try to attack the cancer. It's like a criminal organization that doesn't just avoid the police, but actually recruits police officers to protect their headquarters. The corruption goes deeper still. Cancer cells can produce growth factors that were originally designed to help tissues regenerate after injury. They can activate stem cell programs that normally help organs repair themselves. They can even trigger the formation of new lymphatic vessels, part of your body's drainage and immune surveillance system, that end up providing highways for cancer cells to spread to distant locations. Dr. Harold Varmus, who won the Nobel Prize for his work on cancer genetics, once observed that cancer is like a caricature of normal cellular behavior. All the right processes, but amplified and distorted beyond recognition. The cells divide, which is normal. They move, which is normal. They recruit blood supply, which is normal. They communicate with other cells, which is normal. But they do all of these normal things in abnormal ways, at abnormal times, in abnormal places. This corruption of healing processes helps explain why cancer can be so difficult to detect in its early stages. The tumor isn't just hiding from your body's surveillance systems, it's actively using those systems to appear more legitimate, more necessary, more like something that belongs there. It also explains why some cancer treatments can feel like they're making patients sicker before they make them better. When doctors disrupt the abnormal blood vessel networks that feed tumors, they're also disrupting some of the body's normal healing processes. When they activate the immune system to attack cancer, they risk triggering autoimmune responses against healthy tissue. Fighting cancer often means temporarily sabotaging some of your body's most generous impulses. Its desire to heal, to grow, to repair, to protect itself from excessive immune responses. It's like having to ground your most helpful child, because they've been manipulated into helping the class troublemaker. But understanding this corruption also reveals new therapeutic opportunities. If cancer depends on hijacking normal healing processes, then carefully targeted disruption of those processes can starve tumors without devastating healthy tissue. Anti-angiogenic therapies try to cut off the tumor's blood supply by blocking the signals that recruit new blood vessel growth. Immunotherapy tries to overcome the tumor's deceptive tolerance signals and reactivate immune recognition of cancer cells. Anti-inflammatory approaches try to quiet the chronic inflammatory environment that feeds tumor growth. These approaches acknowledge something profound about cancer treatment. Sometimes the most effective therapy isn't about introducing something foreign to kill the tumor, but about restoring normal regulatory processes that cancer has corrupted. It's about teaching your body to remember how to say no to unreasonable requests for help. It's about helping your immune system see through cancer's deceptive diplomatic credentials. It's about restoring the normal checks and balances that keep healing from becoming harmful. In many ways, treating cancer is about helping your body remember how to be itself again, generous but not gullible, healing but not hijacked, protective but not naive. It's about restoring the wisdom of biological systems that have evolved over millions of years, to distinguish between growth that serves life and growth that threatens it. The betrayal runs deep, but so does the body's capacity for learning, adaptation, and ultimately resistance to deception. Why the immune system often misses cancer. Your immune system is, without exaggeration, one of the most sophisticated security networks ever created. It patrols your body 24 hours a day, 7 days a week, equipped with the biological equivalent of facial recognition software, chemical fingerprinting, and a communication network that would make the CIA envious. It can distinguish between millions of different potential threats, remember encounters from decades ago, and coordinate responses involving billions of cells across your entire body. So why does this incredible system sometimes let cancer slip right past its defenses? The answer lies in a peculiar fact about cancer that we touched on earlier. Cancer cells are not foreign invaders, they're your own cells, carrying your own genetic passport, speaking your body's molecular language fluently. Asking your immune system to recognize and eliminate cancer is like asking airport security to identify terrorists who look exactly like ordinary passengers, carry legitimate identification, and know all the right things to say. But the challenge goes even deeper than that. Imagine you're a security guard at a large office building. Your job is to keep out intruders while allowing employees to go about their work. Now imagine that some of the employees have gradually become corrupt. They're embezzling money, selling company secrets, and undermining the organization from within. But they still have valid ID badges. They still know the right passwords. And they still look like everyone else who works there. That's essentially what your immune system faces with cancer. Dr. William Coley discovered over a century ago that the immune system could sometimes be coaxed into fighting cancer. He noticed that cancer patients who developed severe bacterial infections sometimes saw their tumors shrink. The infections, it seemed, were waking up the immune system and making it more aggressive against all abnormal cells, including cancer cells. This observation led to decades of research into what scientists call immune surveillance, the idea that your immune system is constantly patrolling for cancer cells and eliminating them before they can cause problems. And indeed, this surveillance does work much of the time. Scientists estimate that your immune system probably prevents thousands of potential cancers throughout your lifetime, quietly eliminating abnormal cells before they can establish themselves as tumors. But cancer cells that do manage to grow into detectable tumors have, by definition, already learned to evade immune surveillance. They're the ones that figured out how to stay hidden, how to avoid detection, or how to neutralize immune responses against them. Some cancers master the art of molecular camouflage. They reduce the display of proteins on their surface that would normally identify them as abnormal. It's like removing your name tag and trying to blend into a crowd. If you don't stand out, security might not notice you're not supposed to be there. Other cancers become experts at immune suppression. They produce chemicals that essentially drug the immune cells around them, making them sluggish, confused, or inactive. Imagine if those corrupt employees could somehow put sedatives in the security guards' coffee. They could walk right past with briefcases full of stolen goods, and the guards would be too drowsy to notice or care. Perhaps most insidiously, some cancers learn to exploit the immune system's built-in safety mechanisms. Your immune system has several biological off switches designed to prevent it from becoming overactive and attacking healthy tissue. These regulatory mechanisms are essential. Without them, you'd develop autoimmune diseases, where your immune system destroys your own organs. But cancer cells can learn to activate these off switches, essentially telling immune cells, "Stand down, everything is fine here. No need to investigate further." They wave false flags of immune suppression, convincing regulatory cells to actively protect the tumor from immune attack. Dr. James Allison won the Nobel Prize for discovering one of these off switches, a protein called CTLA4 that acts like a brake pedal on immune cell activation. He realized that cancer cells were essentially pressing this brake pedal to stop immune attacks against them. His breakthrough was developing drugs that could block this braking system, releasing the immune system to attack cancer more aggressively. This discovery launched the field of immunotherapy, but it also revealed how sophisticated cancer's immune evasion strategies really are. Tumors don't just hide from the immune system, they actively manage it, manipulate it, and use it for their own purposes. Some cancers recruit regulatory immune cells that normally prevent autoimmune responses and use them as a protective shield. Others produce chemicals that convert ordinary immune cells into tumor-supporting cells. Still others create what scientists call immune deserts, regions where immune cells simply cannot penetrate or survive. But perhaps the most challenging aspect of cancer's immune evasion is its heterogeneity. Remember that tumors are not uniform masses of identical cells. They're diverse ecosystems where different cancer cell populations may use different immune evasion strategies. Some cells in the tumor might be hiding through camouflage, while others are actively suppressing immune responses, and still others are recruiting immune cells to help them grow. This diversity means that even if an immunotherapy successfully targets one immune evasion mechanism, other cancer cells in the same tumor might be using completely different strategies to avoid detection. It's like trying to catch a criminal organization where different members are using different disguises, different hideouts, and different methods of corruption. The timing of immune responses also matters in ways that favor cancer. Your immune system is designed to respond quickly to acute threats, infections, injuries, foreign substances, but cancer develops slowly, over months or years. By the time a tumor is large enough to detect, cancer cells have had extensive time to train the local immune environment to tolerate their presence. It's like the difference between a burglar who breaks into your house suddenly versus a con artist who gradually moves in, gains your trust, and slowly takes over your property. The sudden burglar triggers all your alarm systems. The gradual con artist might live in your house for years before you realize something's wrong. This gradual immune education helps explain why early detection is so crucial in cancer treatment. The longer cancer cells have to establish themselves and train the local immune environment, the more sophisticated their evasion strategies become. But understanding these evasion mechanisms also reveals new therapeutic opportunities. If cancer succeeds by manipulating immune responses, then restoring normal immune function can be powerfully therapeutic. Checkpoint inhibitor drugs, like those developed by Dr. Allison, work by removing the false off switches that cancer cells use to suppress immune attacks. CAR cell therapy involves removing immune cells from patients, genetically engineering them to better recognize cancer cells, and then returning them to the body as enhanced cancer hunting specialists. Other approaches try to make cancer cells more visible to the immune system by increasing their display of identifying proteins or by attaching molecular wanted posters to their surfaces. Still others work to overcome the immune suppressive environment that tumors create. essentially clearing the chemical fog that cancer uses to confuse immune surveillance. The field of cancer immunotherapy has exploded in recent years, with new approaches being developed that turn the immune system into a more effective cancer-fighting force. But success requires understanding cancer, not just as a growth problem, but as an immune evasion problem, a challenge that demands both biological insight and strategic thinking. Your immune system isn't broken when it fails to eliminate cancer. It's being actively deceived, manipulated, and outmaneuvered by cells that have learned to exploit its most generous and protective instincts. The goal of immunotherapy isn't to create a more aggressive immune system, but to create a wiser one that can see through cancer's deceptions and remember its original mission to protect the body from cellular rebellion. In many ways, this represents a return to Dr. Coley's original insight. Sometimes the best way to fight cancer is not to attack it directly, but to wake up the body's own security forces and help them do what they were originally designed to do. Tumor microenvironments, fortresses of resistance. If you could shrink yourself down to the size of a cell and walk through a tumor, you wouldn't find yourself in the orderly, well-planned neighborhood that characterizes healthy tissue. Instead, you'd discover something more like a post-apocalyptic city, chaotic, hostile, and organized around survival rather than cooperation. This is the tumor microenvironment, and it's one of cancer's most ingenious innovations. Cancer cells don't just rebel against the body's rules, they actively reshape their surroundings to create living spaces that favor their survival and growth. They're not just squatters in the body's neighborhoods, they're urban planners redesigning entire districts to serve their needs. Imagine a crime boss who doesn't just commit crimes, but gradually transforms the entire neighborhood to make crime easier and law enforcement harder. They bribe the police, intimidate witnesses, control the local businesses, and create a culture where their criminal activities seem normal, even necessary. That's essentially what cancer cells do with their local environment. Dr. Bissell, a pioneering researcher in cancer biology, spent decades demonstrating that context matters as much as genetics in cancer development. She showed that cancer cells placed in normal, healthy environments often behave normally, while normal cells placed in tumor-like environments can begin to act like cancer cells. It's not just the cells that matter, it's the entire neighborhood they live in. So what makes a tumor microenvironment so different from normal tissue? First, there's the architecture. In healthy tissue, cells are organized in orderly patterns with clear boundaries between different cell types. There are designated spaces for blood vessels, structural support cells, and various specialized tissue types. Everything has its place, and everything stays in its place. But in tumors, this organization breaks down. Cancer cells bulldoze through normal tissue boundaries, creating chaotic mixtures of cell types. The structural framework that normally provides organization and stability gets dismantled and rebuilt in haphazard ways. It's like a construction site where the workers have thrown out all the blueprints and are just building whatever seems convenient at the moment. The blood supply in tumors tells its own story of chaos. Remember how cancer cells can hijack the body's blood vessel construction system. The result is a vascular network that looks nothing like the elegant, efficient blood vessels in healthy tissue. Tumor blood vessels are leaky, poorly organized, and inefficient. They create areas of the tumor that are well fed alongside areas that are starving for oxygen and nutrients. You might think this poor blood supply would hurt the tumor, but cancer cells have learned to thrive in these harsh conditions. Cells in low oxygen environments become more aggressive, more likely to spread, and more resistant to many types of treatment. It's like how people who grow up in tough neighborhoods often develop survival skills that make them harder to defeat. The chemical environment in tumors is equally hostile. Normal tissue maintains a carefully balanced pH, like a well-maintained swimming pool, but tumors are often acidic, creating a caustic environment that normal cells can't tolerate, but cancer cells have learned to love. This acidity helps cancer cells break down barriers between tissues, making it easier for them to invade surrounding areas. Cancer cells also fill their environment with growth factors, inflammatory signals, and other chemicals that create a pro-tumor atmosphere. It's like they're constantly playing music that makes other cells want to dance to their rhythm. Perhaps most remarkably, tumors recruit and corrupt normal cells to serve their purposes. Healthy cells that find themselves in tumor environments often get reprogrammed to become cancer supporters. Immune cells that should be attacking the tumor instead become tumor bodyguards. Structural cells that should be maintaining tissue organization instead help the tumor spread. Blood vessel cells that should be delivering nutrients efficiently instead create the chaotic vascular networks that tumors prefer. Dr. Douglas Hanahan and Robert Weinberg, in their influential analysis of cancer biology, describe the tumor microenvironment as an enabling environment, a place that doesn't just tolerate cancer but actively promotes it. It's like a corruption that spreads outward from cancer cells, gradually converting normal tissue into tumor-supporting tissue. This microenvironment creates what scientists call niches, specialized microhabitats within the tumor where different cancer cell populations can thrive. Some niches favor cancer stem cells, the small population of tumor cells that can regenerate the entire tumor if given the chance. Other niches favor cells that are particularly good at invading surrounding tissue or surviving in harsh conditions. The result is that tumors become incredibly resilient ecosystems. Even if treatment eliminates most of the cancer cells, the microenvironment they've created can persist, providing a favorable habitat for any surviving cancer cells to regrow the tumor. This is one reason why cancer can recur in the same location even after apparently successful treatment. The tumor microenvironment acts like contaminated soil. Even if you remove all the visible weeds, the environment itself encourages new weeds to grow. Understanding tumor microenvironments has revolutionized how scientists think about cancer treatment. It's not enough to just kill cancer cells, you also need to disrupt the supportive environment they've created. Some newer cancer therapies specifically target the tumor microenvironment. Anti-angiogenic drugs try to normalize the chaotic blood vessel networks in tumors. Drugs that target cancer-associated fibroblasts try to convert tumor-supporting structural cells back into normal tissue-maintaining cells. Approaches that modulate the immune microenvironment try to convert tumor-promoting immune cells back into tumor-fighting immune cells. Other strategies try to make the tumor microenvironment less hospitable to cancer cells. Drugs that reduce tumor acidity make it harder for cancer cells to break down tissue barriers. Treatments that improve oxygen delivery to tumors can make them more vulnerable to radiation therapy and some types of chemotherapy. There's even research into completely reshaping tumor microenvironments, essentially performing urban renewal at the cellular level. Scientists are exploring ways to deliver normal structural cells, normal immune cells, and normal blood vessels into tumors, hoping to gradually convert the chaotic tumor environment back into something more like healthy tissue. The tumor microenvironment also helps explain why different tumors in the same organ can behave so differently. It's not just the genetics of the cancer cells that matter, but the entire ecological system they've created around themselves. Two tumors with similar genetics might create completely different microenvironments, leading to completely different clinical behaviors. This environmental aspect of cancer adds another layer of complexity to treatment, but it also adds new opportunities. If cancer depends on creating and maintaining a supportive microenvironment, then disrupting that environment becomes a powerful therapeutic strategy. It's like realizing that to eliminate organized crime, you don't just need to arrest the criminals, you also need to clean up the neighborhoods they've corrupted, restore legitimate businesses, and rebuild the social structures that make crime less attractive than honest work. The tumor microenvironment represents cancer at its most sophisticated, not just rebelling against the body's rules, but rewriting those rules to create entire districts where rebellion becomes the norm. Understanding these environments is crucial to understanding why cancer is so resilient, so adaptable, and ultimately, so challenging to eliminate completely. But like any environment, tumor microenvironments can be changed, managed, and eventually restored to healthier states. The key is recognizing that cancer isn't just a cellular problem. It's an ecological problem that requires ecological solutions. Diversity within no two cancers are the same. There's a moment that happens in every oncologist's career when they realize something that textbooks can't quite capture. Two patients can walk into their office with what appears to be identical cancer, same organ, same stage, same microscopic appearance, and yet their diseases will behave as differently as two snowflakes melting in different climates. This realization is both humbling and profound. It means that despite all our medical sophistication, despite genetic sequencing and molecular profiling and precision medicine, cancer retains an almost infinite capacity to surprise us, and this diversity isn't a bug in the system. It's a feature that cancer has evolved to exploit. Imagine trying to write a guidebook for a city where every building constantly changes its architecture, every street rearranges itself weekly, and every neighborhood develops its own unique culture and language. That's essentially what cancer researchers face when they try to understand and categorize different cancers. The diversity begins at the most fundamental level, the genetic mutations that drive cancer development. When scientists first began sequencing cancer genomes, They expected to find a relatively small number of key mutations that could explain most cancers. Instead, they discovered what Dr. Berta Vogelstein calls the long tail of cancer genetics. Thousands of different genes that can contribute to cancer development, each mutated in different combinations in different patients. Two women with breast cancer might have tumors that share fewer genetic similarities than either tumor shares with a lung cancer in a completely different patient. It's like discovering that two houses on the same street were built using completely different architectural principles, different materials and different construction techniques. But the diversity goes far beyond genetics. Even within a single tumor, different regions can contain cancer cells with dramatically different characteristics. Scientists call this intratumor heterogeneity, and it's one of cancer's most challenging features. Dr. Charles Swanton, who studies this phenomenon extensively, uses the metaphor of a forest. From a distance, a forest might look uniform, just a mass of green trees, but walk through it, and you'll discover oak groves, pine stands, birch clusters and dozens of different microenvironments, each supporting different communities of plants and animals. Tumors are similar. What looks like a uniform mass of cancer cells from the outside is actually a complex ecosystem containing multiple populations of cancer cells, each with its own genetic signature, its own survival strategies, and its own response to treatment. This means that when doctors take a biopsy, a small sample of tumor tissue, they're essentially looking at one tree and trying to understand the entire forest. The sample might miss entirely different populations of cancer cells that exist in other parts of the tumor. It's like trying to understand the biodiversity of the Amazon rainforest by examining a single branch. The clinical implications of this diversity are staggering. A treatment that successfully targets the cancer cells identified in the biopsy might have no effect on genetically different cancer cells elsewhere in the tumor. Those resistant cells can then multiply and repopulate the tumor, leading to treatment failure and cancer recurrence. Even more challenging is the fact that this diversity isn't static. Cancer cells continue to mutate and evolve throughout the course of the disease, creating new populations with new characteristics. It's like trying to hit a target that's not only moving but constantly changing shape, size, and color. Dr. Cornelia Polyak, who studies breast cancer heterogeneity, discovered that tumors can contain cancer cells in different stages of development, some that are just beginning their transformation from normal cells, others that are intermediate in their development, and still others that are fully transformed and aggressive. It's like finding teenagers, adults and elderly people all living in the same household, each with different needs, different behaviors, and different responses to the same stimuli. This diversity also explains why cancer staging, the system doctors use to describe how advanced a cancer is, can be misleading. Two tumors that appear to be at the same stage based on size and spread might have completely different genetic landscapes, different growth rates, and different likelihoods of responding to treatment. The challenge is compounded by what scientists call phenotypic plasticity, the ability of cancer cells to change their behavior and characteristics in response to environmental pressures. Some cancer cells can switch between different states, like actors who can play multiple roles in the same play. For example, some cancer cells can alternate between a state that favors rapid growth and a state that favors survival under harsh conditions. When chemotherapy creates a hostile environment, these cells might switch to survival mode, becoming dormant and treatment-resistant. When the treatment ends and conditions improve, they can switch back to growth mode and regenerate the tumor. Other cancer cells can change their identity entirely, transforming from one cell type to another to escape targeted treatments. Lung cancer cells, under pressure from targeted therapy, have been observed to transform into small-cell lung cancer, a completely different type of cancer that requires different treatment approaches. This plasticity makes cancer an incredibly adaptable opponent. It's like fighting an enemy who can instantly change uniforms, weapons, and tactics in response to your strategy. The diversity of cancer also extends to how different tumors interact with their hosts. Some cancers are like aggressive invaders, growing rapidly and spreading quickly. Others are more like stealth infiltrators, growing slowly and hiding from immune surveillance for years. Still others are like diplomatic manipulators, recruiting normal cells to support their growth and spread. Even cancers that arise in the same organ can have completely different personalities. Breast cancer can be driven by estrogen, by growth factor receptors, by specific genetic mutations, or by combinations of these and other factors. Each subtype behaves differently, spreads differently, and responds differently to treatment. This diversity initially seemed like an insurmountable obstacle to effective cancer treatment. How can you develop therapies for diseases that are essentially unique to each patient? But the diversity also contains seeds of hope, because if every cancer is different, then every cancer also has unique vulnerabilities. The goal of precision medicine is to identify these individual vulnerabilities and exploit them with targeted therapies. Modern cancer treatment increasingly involves comprehensive molecular profiling of individual tumors, genetic sequencing, protein analysis, and immune system characterization. The goal is to create a detailed molecular portrait of each patient's specific cancer, then match that portrait to the most appropriate treatments. Some hospitals now have molecular tumor boards where teams of specialists review each patient's unique cancer profile and design individualized treatment strategies. It's like having a team of detectives analyze the specific criminal organization in each case and develop customized law enforcement strategies. The diversity of cancer also drives the development of combination therapies designed to target multiple aspects of tumor biology simultaneously. If cancer cells can use multiple survival strategies, then effective treatment must address multiple vulnerabilities at once. Liquid biopsies, blood tests that can detect circulating tumor DNA, are beginning to allow doctors to monitor the genetic evolution of cancers in real time, adjusting treatments as the tumor's genetic landscape changes. Perhaps most importantly, understanding cancer diversity has led to a more humble and flexible approach to treatment. Instead of expecting one-size-fits-all solutions, oncologists are learning to think like ecologists, naturalists, and evolutionary biologists, professionals who understand that diversity is not a problem to be solved, but a fundamental feature of life that must be understood and worked with rather than against. The infinite variety of cancer is indeed one of its greatest strengths, but it's also what makes each victory against cancer meaningful. Each successful treatment a small miracle of scientific insight, matching biological reality. In the end, cancer's diversity reminds us that medicine, at its best, is not about applying universal solutions to standard problems. It's about understanding the unique story that each disease tells in each patient, and finding ways to help write a better ending to that story. Metastasis: how cancer spreads and hides. There's a moment in every cancer journey that changes everything. The moment when doctors discover that the disease has spread beyond its original location. Suddenly, what might have been a localized problem requiring localized treatment becomes a systemic challenge involving the entire body. This process, called metastasis, represents cancer at its most successful and most terrifying. Metastasis is not just cancer growing larger, it's cancer learning to travel, to survive in foreign environments, and to establish new colonies in distant parts of the body. It's the difference between a house fire and a forest fire, between a local rebellion and a revolution that spreads across continents. To understand how extraordinary this process really is, consider what cancer cells must accomplish to metastasize successfully. They must break away from their original tumor, survive in the bloodstream or lymphatic system, travel to distant organs, exit the circulation, survive in completely foreign tissue environments, and establish new tumors that can grow and thrive in their new locations. It's like asking someone to escape from prison, survive in the wilderness, travel to a foreign country without documentation, blend in with a completely different culture, and then establish a successful new life there. The fact that cancer cells can do this at all is remarkable. The fact that they do it regularly is terrifying. Dr. Isaiah Fidler, who spent decades studying metastasis, discovered that the process is incredibly inefficient. Of the millions of cancer cells that break away from primary tumors and enter the circulation, fewer than 0.1% successfully establish metastatic colonies. The vast majority die during the journey, or fail to adapt to their new environments. But here's the unsettling part. Even this tiny success rate is enough. A single successful metastatic cell can potentially give rise to a new tumor containing billions of cells. It's like biological gambling where the odds are terrible, but the stakes are enormous, and cancer keeps playing until it wins. The metastatic process begins with something called epithelial mesenchymal transition, a mouthful of scientific terminology that describes something quite elegant and sinister. Epithelial cells are the well-behaved citizens of the body. They stay put, maintain strong connections with their neighbors, and respect tissue boundaries. Mesenchymal cells are the wanderers. They can move freely, survive independently, and ignore the social rules that keep epithelial cells in line. Cancer cells can learn to switch between these two states, essentially transforming from settled citizens into nomadic explorers when they need to spread. It's like discovering that mild-mannered office workers can instantly transform into survivalist adventurers whenever they want to leave their jobs and explore new territories. Once cancer cells enter the bloodstream, they face a hostile environment that would kill most normal cells within minutes. The blood is full of immune cells actively hunting for anything that doesn't belong there. The physical forces of circulation can tear fragile cells apart. The chemistry of blood is completely different from the tissue environments where most cells are designed to live. But metastatic cancer cells have developed remarkable survival strategies. Some travel in clusters, protecting each other like wolves in a pack. Others coat themselves with platelets, the blood cells responsible for clotting, essentially disguising themselves as innocent blood clots. Still others seem to enter a kind of suspended animation, reducing their metabolic activity to almost nothing until they reach a more hospitable environment. The process becomes even more complex when cancer cells try to exit the circulation and establish new tumors. Different organs have different defenses against invasion, different nutritional environments, and different immune surveillance systems. Some primary tumors actually send out advanced signals that prepare distant organs for metastatic colonization. They recruit normal cells in distant locations to create what scientists call pre-metastatic niches, specialized environments that will be welcoming to cancer cells when they arrive. It's like sending advanced teams to foreign countries to establish safe houses and support networks before the main invasion begins. These pre-metastatic niches can exist for months or years before the first cancer cells arrive, like hotel rooms reserved for guests who haven't yet made their travel plans. When metastatic cells finally do arrive, they find environments that have already been modified to support their survival and growth. Perhaps most troubling is the phenomenon of dormant metastases. Cancer cells can travel to distant organs and remain dormant, essentially sleeping for years, or even decades before suddenly awakening and growing into detectable tumors. This is why cancer can recur years after apparently successful treatment of the primary tumor. These dormant cells are like sleeper agents planted deep in enemy territory. They're not actively growing or dividing, which makes them nearly invisible to both the immune system and to treatments that target rapidly dividing cells. They wait, in a state of suspended animation, until conditions become favorable for growth. What triggers dormant metastases to awaken is still largely mysterious. Sometimes it seems to be hormonal changes, like those that occur during pregnancy or menopause. Sometimes it appears to be other illnesses or stresses that alter the body's internal environment. Sometimes it seems to be completely random. This dormancy phenomenon helps explain one of cancer medicine's most cruel ironies. Patients can be declared cancer-free after successful treatment, only to have the disease return years later in completely different organs. The cancer wasn't really gone, it was just sleeping. Modern cancer treatment increasingly recognizes metastasis as a separate problem, requiring separate solutions. Anti-metastatic therapies try to prevent cancer cells from successfully completing the complex process of establishing new tumors in distant organs. Some approaches try to prevent cancer cells from breaking away from primary tumors in the first place. Others try to make the bloodstream more hostile to circulating cancer cells. Still others try to prevent cancer cells from successfully establishing themselves in distant organs, or try to keep metastatic cells dormant rather than allowing them to grow into tumors. Liquid biopsies can sometimes detect circulating tumor cells or tumor DNA in the bloodstream, potentially allowing doctors to identify and treat metastatic disease before it becomes detectable by conventional imaging. Understanding metastases has also changed how doctors think about cancer staging and treatment. The presence or absence of metastatic disease is often more important for determining prognosis than the size or characteristics of the primary tumor. A small primary tumor that is already metastasized is generally much more dangerous than a large primary tumor that has remained localized. The biology of metastasis reveals something profound about cancer. It's not just a disease of uncontrolled growth, but a disease of inappropriate movement and adaptation. Cancer cells don't just forget how to stop dividing, they learn how to survive in places where they were never supposed to exist. In many ways, metastasis represents evolution in fast forward, as cancer cells rapidly develop the ability to survive in multiple different environments within a single organism. It's like watching a species adapt to different continents within the span of months rather than millennia. But understanding this process also reveals new opportunities for intervention. If metastasis requires cancer cells to successfully complete multiple complex steps, then disrupting any one of those steps can prevent the process from succeeding. The war against metastasis is really a war against cancer's mobility, adaptability, and patience. It requires strategies that can target cancer not just where it is but where it might go, and not just when it's growing but when it's hiding and waiting for better opportunities. It's a reminder that cancer is not just a local problem that gets out of hand, but a systemic disease that can use the body's own transportation and communication networks against it. Fighting metastasis means thinking like cancer thinks, strategically, patiently, and always several steps ahead. Cancer stem cells, the immortals inside tumors. Hidden within every tumor lies a small population of cells that scientists have come to regard with a mixture of fascination and dread. These are cancer stem cells, the cellular masterminds that can single-handedly regenerate entire tumors even after treatments that eliminate 99.9% of the cancer. They're like the queen in a chess game or the generals in an army. Take out all the other pieces but leave them alive, and they can rebuild everything you've destroyed. The discovery of cancer stem cells fundamentally changed how scientists think about tumor biology. For decades, researchers assumed that all cancer cells within a tumor were essentially equivalent, that any cancer cell could grow into a new tumor given the right conditions. But it turns out that tumors, like healthy tissues, have a hierarchy. Not all cancer cells are created equal. Dr. John Dick first demonstrated this hierarchy in leukemia in the 1990s. He showed that only a tiny fraction of leukemia cells, less than 1% in many cases, could actually generate new tumors when transplanted into laboratory animals. The vast majority of cancer cells, despite being malignant, lacked this regenerative potential. They could divide a few times, but they couldn't sustain long-term tumor growth. This was a revolutionary discovery. It meant that tumors weren't just chaotic masses of identical cancer cells, but organized societies with leadership structures. The cancer stem cells were the leaders, small in number but essential for the survival and growth of the entire tumor. Imagine a criminal organization where most members can commit crimes but only a few have the knowledge and connections necessary to rebuild the entire organization if it's dismantled by law enforcement. Those few key figures are the ones that really matter for the long-term survival of the criminal enterprise. Cancer stem cells have several characteristics that make them particularly dangerous. First, they divide slowly and infrequently compared to other cancer cells. This is actually an advantage, because many cancer treatments, chemotherapy, radiation therapy, are designed to target rapidly dividing cells. Cancer stem cells can essentially sleep through treatments that devastate the rest of the tumor. It's like how the wisest generals in a war often stay far from the front lines, letting their soldiers do the fighting while they preserve themselves to rebuild the army, if necessary. Second, cancer stem cells are incredibly resistant to cellular stress. They have enhanced DNA repair systems that help them survive radiation damage. They have more efficient mechanisms for expelling toxic drugs from their cells. They're better at surviving in harsh conditions like low oxygen or limited nutrients. Dr. Max Witcher, who has spent decades studying breast cancer stem cells, describes them as the ultimate survivors in the tumor ecosystem. While other cancer cells might die when conditions become hostile, cancer stem cells hunker down and wait for better times. Third, cancer stem cells are masters of asymmetric division. When they do divide, they don't necessarily create two identical daughter cells. Instead, they often create one new stem cell, maintaining the stem cell population, and one more differentiated cancer cell expanding the tumor mass. It's like having a factory where the machinery can both produce products and reproduce itself. This asymmetric division means that cancer stem cells can simultaneously maintain themselves and generate the bulk of the tumor. They're both the root and the trunk of the cancer tree. Perhaps most troubling, cancer stem cells appear to be the source of metastatic spread. When tumors metastasize to distant organs, it's often cancer stem cells that make the journey, survive in the new environment, and establish new tumor colonies. The more differentiated cancer cells, despite being more numerous, seem to lack the adaptability necessary for successful metastasis. This helps explain why metastatic disease is so much more dangerous than localized cancer. Metastases aren't just cancer that has spread. They're cancer that has been spread by the most dangerous and adaptable cells in the original tumor. Cancer stem cells also help explain the phenomenon of tumor dormancy. These cells can remain quiescent, essentially sleeping for years or decades before suddenly awakening and regenerating tumor growth. They're like seeds that can remain viable in harsh conditions for extended periods before sprouting when conditions improve. The existence of cancer stem cells has profound implications for cancer treatment. Traditional approaches that focus on shrinking tumors by killing the maximum number of cancer cells might be missing the most important targets. If you eliminate 99% of a tumor but leave the cancer stem cells intact, the tumor can potentially regenerate completely. It's like trying to eliminate a weed by cutting off all the visible parts while leaving the root system intact. The weed will likely grow back, often stronger than before because it now has less competition for resources. This realization has sparked intense interest in developing treatments that specifically target cancer stem cells, but these cells present unique therapeutic challenges. Because they divide slowly, they're resistant to many conventional treatments. Because they're metabolically different from other cancer cells, they might not respond to targeted therapies designed for the bulk tumor population. Some researchers are exploring ways to force cancer stem cells out of their quiescent state, making them more vulnerable to conventional treatments. Others are looking for ways to disrupt the specialized niches where cancer stem cells hide and maintain themselves. Still others are trying to identify unique molecular features of cancer stem cells that could serve as therapeutic targets. Dr. Piyush Gupta and his colleagues have discovered that cancer stem cells can switch back and forth between stem-like and more differentiated states. This plasticity means that even if you successfully eliminate all the cancer stem cells in a tumor, some of the remaining cancer cells might be able to transform back into stem cells and replenish the stem cell population. It's like fighting an enemy, where ordinary soldiers can suddenly promote themselves to general rank when all the real generals have been eliminated. This plasticity makes cancer stem cells an even more elusive target. The cancer stem cell model also helps explain why some cancers are more aggressive than others. Tumors with higher proportions of cancer stem cells tend to be more likely to recur after treatment, more likely to metastasize, and more resistant to therapy. It's not just the total number of cancer cells that matters, but the quality and characteristics of those cells. However, the cancer stem cell model is still evolving, and not all scientists agree on its implications. Some argue that the stem cell hierarchy in tumors is more flexible than originally thought, with cells able to switch roles depending on environmental conditions. Others suggest that the stem cell model, while useful, might be an oversimplification of tumor biology. What's clear is that cancer stem cells represent one of the most sophisticated aspects of tumor biology. They're like the deep state of cancer. Small, hidden, persistent, and incredibly influential despite their small numbers. Understanding cancer stem cells has led to a more nuanced approach to cancer treatment. Instead of just trying to shrink tumors, oncologists are increasingly focused on eliminating the cells that can regenerate tumors. Instead of just measuring treatment success by tumor size reduction, researchers are looking for ways to assess whether cancer stem cells have been eliminated. Some of the most promising new cancer treatments specifically target cancer stem cell populations. These approaches recognize that winning the war against cancer might require not just winning battles against tumor bulk, but eliminating the cellular leadership that can rebuild the enemy forces. The cancer stem cell story is still being written, but it's already clear that these remarkable cells represent both one of cancer's greatest weapons and one of medicine's most important targets. They remind us that cancer is not just about cells growing where they shouldn't, but about cells that have learned to survive, adapt, and rebuild under the most challenging conditions imaginable. In many ways, cancer stem cells embody everything that makes cancer so formidable. They're patient, resilient, adaptable, and seemingly immortal. But they also represent hope, because understanding them gives us new ways to think about how to achieve true, lasting victories against cancer. Escape artists. How cancer outsmarts chemotherapy. There's a moment that haunts every oncologist, the moment when a treatment that was working beautifully suddenly stops working. The patient's scans show tumor shrinkage, blood markers improve, symptoms resolve, everyone celebrates. Then months later the cancer returns, and this time, the same treatment that worked so well before has no effect at all. The cancer has learned to resist. This isn't a failure of medicine or a lack of trying hard enough. This is cancer demonstrating one of its most sophisticated survival skills, the ability to develop resistance to almost any treatment we can devise. And it happens not in years or decades, but often in months, through processes that would make a master escape artist envious. To understand how cancer develops chemotherapy resistance, imagine a city under siege. The attacking army uses a powerful new weapon that's devastatingly effective at first, destroying most of the city's defenses and eliminating most of the population. But among the survivors are a few individuals who, through luck or genetics or sheer determination, find ways to protect themselves from this new weapon. These survivors don't just hide and wait for the siege to end. They study the weapon, learn its weaknesses, develop countermeasures, and teach these survival skills to others. When the siege lifts, and the city begins to rebuild, It's populated entirely by people who are immune to the weapon that once threatened to destroy everyone. That's essentially what happens when cancer develops chemotherapy resistance. The process often begins with what scientists call intrinsic resistance. Some cancer cells within a tumor are naturally less sensitive to chemotherapy from the very beginning. This isn't because they're trying to be difficult. It's because cancer cells, like all cells, have natural variation in their characteristics. Some happen to have genetic mutations that make them better at surviving chemical assault. Dr. Goldie and Coldman, two Canadian researchers, developed a mathematical model in the 1970s that predicted this phenomenon. They calculated that even in a tumor that appears highly sensitive to chemotherapy, there are likely to be some cancer cells with natural resistance. The larger the tumor, the more likely it is to contain resistant cells. This means that from the moment treatment begins, there's a race between the chemotherapy-killing sensitive cancer cells and the resistant cells multiplying to replace them. If the treatment can eliminate all the sensitive cells before the resistant ones take over, the patient might be cured. But if resistant cells survive and proliferate, the tumor will eventually become dominated by chemotherapy-resistant cancer cells. But cancer's resistance mechanisms go far beyond this natural selection process. Cancer cells can actively develop new resistance strategies in response to treatment pressure, like prisoners who learn to pick locks after being incarcerated. One of the most common resistance mechanisms involves what scientists call multi-drug resistance pumps, molecular machines that cancer cells use to expel chemotherapy drugs before they can do damage. It's like installing sump pumps that bail out water as fast as it comes in during a flood. These pumps weren't originally designed to cause cancer treatment problems. They're normal cellular machinery that helps cells get rid of toxic substances, a basic survival mechanism that's been around for millions of years. But cancer cells can overexpress these pumps, essentially turning up the volume on their detoxification systems until they can expel even powerful chemotherapy drugs. Dr. Susan Bates, who has studied multi-drug resistance extensively, describes it as cancer cells learning to become cellular whodunit masters of escaping from chemical restraints that should be fatal. Other cancer cells develop resistance by learning to repair DNA damage more efficiently. Most chemotherapy drugs work by damaging cancer cell DNA so severely that the cells die, but some cancer cells respond to this assault by upgrading their DNA repair systems, essentially becoming better at fixing the damage that chemotherapy causes. It's like a city that responds to bombing by becoming exceptionally good at rapid reconstruction. The bombs still cause damage, but the damage gets repaired so quickly that the overall effect is minimal. Still, other cancer cells develop resistance by changing their metabolism, the chemical processes that keep them alive. Some chemotherapy drugs work by interfering with specific metabolic pathways that cancer cells depend on. But cancer cells can learn to use alternative pathways, like a city that responds to the destruction of its main power plant by quickly building backup generators. Perhaps most remarkably, some cancer cells develop resistance by changing their fundamental identity. Under pressure from targeted therapy, lung cancer cells have been observed to transform into completely different types of cancer cells that aren't affected by the original treatment. It's like a criminal who responds to a manhunt by undergoing plastic surgery and assuming an entirely new identity. The speed at which cancer cells can develop these resistance mechanisms is often startling. Genetic analysis of tumors before and after treatment sometimes reveals dozens or hundreds of new mutations that appeared during the course of therapy. It's evolution under extreme pressure, compressed into timescales that would normally require thousands of generations. Dr. Charles Swanton's research has shown that chemotherapy itself can actually accelerate the development of resistance by creating selective pressure that favors the survival of the most adaptable cancer cells. It's like using pesticides in a way that kills most insects, but inadvertently breeds super insects that are resistant to all available treatments. This evolutionary pressure explains why combination chemotherapy using multiple drugs simultaneously is often more effective than sequential single-drug treatments. If cancer cells need to develop resistance to multiple different mechanisms simultaneously, the challenge becomes much more difficult. It's like trying to become simultaneously bulletproof, fireproof, and explosion-proof rather than just developing one type of protection. But even combination therapies face the challenge of cancer's evolutionary creativity. Some tumors develop collateral sensitivity, resistance to one drug that accidentally makes them more sensitive to another drug. Others develop cross-resistance, resistance mechanisms that protect against multiple different drugs simultaneously. The tumor microenvironment also plays a role in chemotherapy resistance. Some areas within tumors have poor blood supply, which means chemotherapy drugs don't reach therapeutic concentrations in those regions. Cancer cells in these poorly perfused areas can survive treatment not because they're genetically resistant, but because they're geographically protected. Other areas within tumors have chemical environments that inactivate chemotherapy drugs or make them less effective. It's like having safe rooms within a building that are designed to protect against specific types of attack. Understanding chemotherapy resistance has led to new treatment strategies that try to outsmart cancer's escape mechanisms. Some approaches use resistance modifiers, drugs that specifically target the mechanisms cancer cells use to resist chemotherapy. Others use metronomic dosing schedules that deliver chemotherapy continuously at low doses, trying to prevent cancer cells from recovering between treatments. Adaptive therapy represents a particularly innovative approach to resistance. Instead of trying to eliminate all cancer cells as quickly as possible, adaptive therapy tries to maintain a stable population of treatment-sensitive cancer cells that can compete with resistant cells and prevent them from taking over the tumor. It's like maintaining a balance between different criminal factions in a city, preventing any one group from becoming dominant rather than trying to eliminate crime entirely. Liquid biopsies can sometimes detect the development of resistance mechanisms before they become clinically apparent, potentially allowing doctors to change treatment strategies before resistance becomes a major problem. The story of chemotherapy resistance is ultimately a story about the incredible adaptability of life itself. Cancer cells, under the pressure of treatments designed to kill them, demonstrate evolutionary creativity that's both terrifying and remarkable. But understanding these resistance mechanisms also provides hope. Every escape route that cancer discovers is a route that can potentially be blocked. Every resistance mechanism that develops is a mechanism that can potentially be targeted. The key is staying one step ahead in an evolutionary arms race, where the stakes couldn't be higher. It requires not just developing new treatments, but anticipating how cancer will adapt to those treatments and preparing countermeasures in advance. In many ways, the battle against chemotherapy resistance is like playing chess against an opponent who can learn from every move you make and adapt their strategy accordingly. The challenge is formidable, but it's not insurmountable. It just requires thinking several moves ahead and always being prepared for cancer's next clever escape attempt. Resistance reloaded. Why some cancers come back stronger. There's a phenomenon in cancer medicine that seems to defy logic. Sometimes after apparently successful treatment, cancer doesn't just return, it comes back more aggressive, more treatment-resistant and more dangerous than it was originally. It's as if the treatment that initially worked so well somehow taught the cancer how to be a more formidable opponent. This isn't medical failure or bad luck. This is cancer demonstrating one of its most unsettling characteristics: the ability to learn from adversity and emerge stronger. Like a villain in a superhero movie, who returns in the sequel with new powers specifically designed to counter the hero's previous victory. Cancer can use the experience of being treated as a training ground for becoming more dangerous. The process often begins during the initial treatment. While chemotherapy or radiation therapy is eliminating the majority of cancer cells, it's also creating an intensely hostile environment that serves as a kind of boot camp for cellular survival. The cancer cells that survive this training are, by definition, the toughest, most adaptable, and most resilient cells in the original tumor. Dr. Robert Gatenby, an evolutionary biologist turned cancer researcher, describes this as treatment-induced selection pressure. Just as harsh environmental conditions in nature select for the strongest and most adaptable organisms, harsh cancer treatments select for the most survival-oriented cancer cells. But the selection process goes beyond just picking the naturally resistant cells from the original tumor. The treatment environment actually induces new mutations and adaptations that wouldn't have occurred otherwise. It's like an extreme training program that doesn't just identify the naturally talented athletes, but actually transforms ordinary participants into superhuman performers. Radiation therapy, for example, damages DNA in cancer cells with the goal of causing so much damage that the cells die. But some cells manage to repair this damage, and in the process of repair they sometimes acquire new mutations that make them more aggressive or more treatment-resistant. The treatment that was meant to kill them instead teaches them new survival skills. Chemotherapy can have similar effects. The cellular stress caused by chemotherapeutic drugs can activate survival pathways in cancer cells that were previously dormant. It's like the difference between a person who's never faced adversity and a person who's been through multiple crises. The latter often develops resilience and problem-solving skills that the former lacks. Dr. Shauna Vasco's research has shown that some cancer cells surviving chemotherapy develop what she calls adaptive stress responses, cellular programs that make them better at surviving not just the original treatment, but a wide range of other hostile conditions. They become generalist survivors rather than specialists. These adapted cells often have enhanced DNA repair capabilities, more efficient mechanisms for expelling toxic drugs, better ability to survive in low-oxygen environments, and improved resistance to immune system attacks. It's like developing a comprehensive survival skill set, rather than just learning to avoid one specific threat. Perhaps most troubling is the phenomenon of treatment-induced stemness. Some cancer cells respond to treatment pressure by becoming more stem-like, developing the characteristics of cancer stem cells that we discussed earlier. These treatment-induced stem cells can be even more dangerous than naturally occurring cancer stem cells because they've been battle-tested under extreme conditions. The tumor microenvironment also plays a role in creating more dangerous recurrent cancers. Treatment can damage normal cells and tissues surrounding the tumor, creating inflammatory conditions that actually promote cancer growth and aggression. It's like urban warfare that destroys civilian infrastructure and creates conditions that favor guerrilla fighters over conventional forces. Some treatments can also disrupt the normal immune surveillance in the treated area, creating immune deserts where cancer cells can grow without immune system interference. The treatment that was meant to eliminate cancer inadvertently creates safe havens where surviving cancer cells can regroup and multiply. Dr. Jingmei Mao has documented cases where radiation therapy intended to eliminate brain tumors instead created microenvironments that favored the growth of more aggressive cancer stems. 2025. We can edit genes. We've mapped the human genome. We carry supercomputers in our pockets. Yet cancer remains this persistent shadow in medicine. This puzzle that seems to grow more complex the closer we look. Maybe you've known someone who fought this battle. Maybe you've wondered why some people beat it while others don't. Maybe you've asked yourself why something so small, just cells behaving badly, can be so impossibly difficult to stop. Tonight, we're going to explore that question together. Targeted therapies versus targetless mutations. Imagine you're a detective trying to catch a criminal who changes their appearance, name, and methods every few days. Traditional cancer treatments were like casting a wide net. Chemotherapy and radiation would attack rapidly dividing cells, hoping to catch the cancer along with healthy tissues. It was effective, but like using a sledgehammer when you need a scalpel. Then came targeted therapies, and for a moment it felt like we had found cancer's fingerprints. Scientists discovered that many cancers had specific molecular signatures, particular proteins or genes that drove their growth. It was like finding out that our shape-shifting criminal always wore the same distinctive watch. We could design drugs that would recognize that watch and stop the criminal in their tracks. Herceptin, for instance, targets a protein called HER2 in certain breast cancers. Glivec revolutionized treatment for chronic myeloid leukemia by targeting a specific protein fusion that drives the disease. These drugs were miracles for many patients. Precision strikes instead of carpet bombing. But cancer, it turns out, is a master of disguise, with an entire wardrobe of watches. The challenge lies in something scientists call tumor heterogeneity. Picture a single tumor not as one entity, but as a bustling city filled with different neighborhoods. Some cancer cells might indeed wear that distinctive HER2 watch, but others in the same tumor might be wearing completely different accessories. When we target the HER2 neighborhood, the other districts simply expand to fill the space. Even more cunning is cancer's ability to develop resistance. It's as if our criminal, after being caught once for wearing that watch, learns to take it off. Cancer cells that survive targeted therapy often develop new mutations that make them immune to the same treatment. They might produce proteins that pump the drug out of the cell, or they might find alternative pathways to grow and divide. Like a criminal who learns to pick locks when their usual crowbar stops working. This evolutionary pressure is relentless. Every treatment we deploy acts like a selective force, killing off the vulnerable cancer cells but leaving behind the resistant ones to multiply. It's Darwin's theory, playing out in fast forward within our own bodies. The cancer cells that survive our best treatments are, by definition, the ones that have learned to evade them. Scientists have discovered that some tumors contain billions of cells, each with subtle genetic differences. That's billions of potential escape routes from any single targeted therapy. Some researchers describe it as trying to drain an ocean with a teacup. Even our most sophisticated targeted drugs can only address a fraction of cancer's complexity at any given moment. The irony is both beautiful and terrifying. The same genetic flexibility that allows life to adapt and survive is what makes cancer so incredibly difficult to eliminate. Cancer cells hijack the fundamental machinery of evolution, using it against us in the most intimate battlefield imaginable, our own bodies. And that brings us to perhaps one of the most promising developments in cancer treatment, immunotherapy. A new hope with old challenges. Think about the last time you had a cold. Your body's immune system, that remarkable internal army, recognized the invading virus and mounted a defense. Fever brought up your internal temperature to make life uncomfortable for the invaders. White blood cells memorized the enemy's face so they could recognize and destroy it faster next time. Your immune system is constantly patrolling, identifying what belongs and what doesn't. Like the world's most sophisticated security system. So why doesn't this incredible defense system stop cancer? The answer lies in cancer's most insidious trick. It learned to wear the uniform of the good guys. Cancer cells arise from our own healthy cells, so they carry many of the same molecular identification cards that tell our immune system, I belong here. It's like a spy who has not only infiltrated the organization, but holds all the right credentials and knows all the right passwords. Immunotherapy represents our attempt to remove cancer's disguise or to enhance our immune system's vision, so it can spot the imposter. Some immunotherapies work like giving our security guards better glasses. They help immune cells recognize cancer's subtle differences from healthy tissue. Others work like removing the do not disturb signs that cancer cells hang on their doors to avoid immune detection. The breakthrough came with understanding immune checkpoints, those molecular breaks that prevent our immune system from attacking our own healthy tissues. Cancer cleverly learned to press these breaks, essentially telling immune cells, nothing to see here, move along. Drugs like pembrolizumab and nivolumab work by releasing these breaks, allowing our immune system to attack cancer with renewed vigor. For some patients, the results have been miraculous. Melanomas that had spread throughout the body have completely disappeared. Lung cancers that were considered terminal have shrunk to undetectable levels. The immune system, once unleashed, can be more thorough and persistent than any drug we've ever created. But immunotherapy has revealed new complexities. Our immune system is like a finely tuned orchestra. Every instrument must play in harmony. When we remove the brakes from immune function, sometimes the orchestra plays too loudly, attacking healthy tissues and causing severe side effects. Patients might develop inflammation in their lungs, liver, or intestines, as their supercharged immune system loses the ability to distinguish friend from foe. More puzzling is why immunotherapy works spectacularly for some patients, but not at all for others. Two people with seemingly identical cancers might have completely different responses. One person's tumor might be what scientists call hot, filled with immune cells ready to attack once the brakes are released. Another person's tumor might be cold, a desert where immune cells rarely venture, no matter how much we enhance their vision or remove their restraints. The tumor microenvironment, that complex ecosystem surrounding cancer cells, plays a crucial role. Some tumors create hostile neighborhoods that immune cells can't penetrate, like fortresses surrounded by moats filled with chemical signals that repel our internal army. Other tumors recruit corrupted immune cells that actually help cancer grow and spread, like security guards who have been bribed to look the other way. Scientists are now working to understand why some immune systems remain vigilant while others seem to fall asleep on the job. They're discovering that factors like age, genetics, previous infections, even the bacteria living in our gut, all influence how well immunotherapy might work. It's as if each person's immune system speaks a slightly different language, and we're still learning how to translate our treatments for each individual dialect. Interestingly, this brings us to another crucial element in cancer's complexity. Timing is everything. The window for effective treatment. Imagine trying to stop a wildfire. In the first few minutes, a single firefighter with a garden hose might contain it. Wait an hour, and you need a fire truck. Wait a day, and you need an entire fleet of aircraft dropping fire retardant. Cancer follows a similar progression. The window of opportunity for effective treatment often depends on catching it before it has had time to establish its defenses and spread its influence. Early-stage cancers are often like that initial spark, They haven't yet learned all their tricks, haven't developed extensive resistance mechanisms, and haven't recruited the surrounding healthy tissue to aid in their growth. Surgery can often remove them completely, radiation can eliminate any remaining cells, and the patient returns to health, as if the cancer had never existed. But cancer is a patient adversary. Many cancers grow silently for months, or even years before causing symptoms. By the time a person feels a lump, experiences pain or notices changes in their body, the cancer may have already established multiple strongholds, developed sophisticated survival mechanisms, and begun the complex process of metastasis, spreading to distant parts of the body. Metastatic cancer represents a fundamentally different challenge. It's no longer a localized problem but a systemic one. Cancer cells that have successfully traveled through the bloodstream or lymphatic system to establish new colonies have already proven themselves to be the most adaptable and resilient members of the cancer population. They're like the most successful explorers from a civilization, carrying with them all the knowledge and tools needed to establish new settlements in foreign territories. The timing of treatment also matters in more subtle ways. Cancer cells don't all divide at the same rate or at the same time. Some are actively growing and dividing, making them vulnerable to certain treatments. Others enter a dormant state, essentially hibernating until conditions improve. It's like trying to catch fish, when some are actively swimming while others are hiding motionless in the deepest parts of the lake. Chemotherapy and radiation primarily target actively dividing cells. Cancer cells that are dormant during treatment can survive like seeds waiting for spring, ready to resume growth once the harsh treatment winter has passed. This is why cancer treatment often requires multiple cycles. We're trying to catch cancer cells during different phases of their life cycle. The concept of cancer stem cells adds another layer to this timing puzzle. These are cancer cells that behave like the master gardeners of a tumor, capable of generating all the different types of cancer cells within a tumor. They often divide slowly and resist standard treatments, hiding like seeds in the soil, while the more visible parts of the cancer are destroyed. Even if treatment eliminates 99% of cancer cells, if these stem cells survive, they can regenerate the entire tumor over time. Scientists have also discovered that the time of day when treatment is given can matter. Our bodies run on circadian rhythms, internal clocks that influence everything from hormone production to cell division. Some cancer treatments work better when given at specific times of day, when normal cells are less active, but cancer cells remain vulnerable. It's as if we're learning to fight cancer, not just with the right weapons, but at the right moment when our target is most exposed. The psychological dimension of timing cannot be ignored either. The window between diagnosis and treatment can feel endless to patients and families. Days turn into anxious weeks as tests are run, specialists consulted and treatment plans developed. Yet this apparent delay often represents crucial preparation, gathering intelligence about the specific type of cancer, its genetic characteristics, and the patient's overall health to customize the most effective approach. This personalized approach leads us to consider the deeper biological foundations of cancer's resilience. The role of genetics and epigenetics in cancer's power. Picture your DNA as an enormous library containing the instructions for building and maintaining your body. Every cell contains the same complete library, but different cells read different books. A liver cell focuses on the metabolism manuals, while ignoring the books about producing tears. A heart cell studies the rhythmic contraction guides, while the neuroscience sections remain closed. Cancer represents what happens when the library's organizational system breaks down. Books that should remain closed get opened. Others that should be regularly consulted get locked away. The careful order that makes cellular life possible dissolves into chaos, and cells begin following conflicting and dangerous instructions. Some people inherit libraries with certain books already damaged. These are the genetic mutations passed down through families. BRCA1 and BRCA2 mutations, for instance, are like having damaged books about DNA repair. When these crucial repair manuals are faulty, other books in the library are more likely to become corrupted over time, leading to increased cancer risk. But inherited genetic mutations account for only about 5 to 10% of cancers. The vast majority arise from damage that accumulates during our lifetime, what scientists call somatic mutations. These are like books in the library that become damaged through use, age, or environmental factors. Smoking introduces chemicals that randomly tear pages from important books, Ultraviolet radiation from the sun causes specific types of damage to the DNA instruction manuals. Even normal aging leads to copying errors as cells divide, like typos accumulating in books that have been reprinted too many times. What makes cancer particularly insidious is that it typically requires multiple damaged books before cells become malignant. Scientists describe this as the multi-hit hypothesis. A single damaged gene rarely causes cancer on its own. It's more like a series of safety systems that must fail before disaster occurs. One broken gene might make a cell grow slightly faster than normal. Another might prevent it from properly repairing DNA damage. A third might disable the cell's built-in suicide mechanism that normally eliminates potentially dangerous cells. This is why cancer becomes more common with age. It's not just that we've had more time for DNA damage to accumulate, it's that we've had more opportunities for multiple safety systems to fail in the same cell. Young people can develop cancer, but it often requires either inherited genetic vulnerabilities or exposure to particularly potent DNA-damaging agents. The discovery of epigenetics added another layer to our understanding of cancer's genetic complexity. While genetics is about the actual text in our DNA library books, epigenetics is about which books are accessible and which are kept on locked shelves. Epigenetic changes don't alter the DNA sequence itself, but control which genes are turned on or off. Think of epigenetic modifications as bookmarks, sticky notes, and locks placed throughout the library. Some bookmarks keep important books open, so the cell can easily access crucial instructions. Other modifications act like locks, keeping potentially dangerous books sealed. Cancer can arise not just from damaged books, but from misplaced bookmarks and broken locks. Environmental factors can influence these epigenetic marks. Diet, stress, exercise, and exposure to chemicals can all affect which genetic books remain accessible to our cells. Some of these epigenetic changes can even be passed down to future generations, like library organizational systems, inherited along with the books themselves. One of the most important discoveries in cancer genetics has been understanding tumor suppressor genes, the books in our genetic library that contain instructions for preventing cancer. The p53 gene, often called the guardian of the genome, produces a protein that monitors cells for DNA damage and either repairs the damage or triggers cell death if the damage is too severe. When p53 is mutated and non-functional, it's like removing the head librarian who normally prevents damaged books from being used. Oncogenes represent the opposite problem. These are genetic instructions that, when functioning normally, help control cell growth and division. But when oncogenes become overactive due to mutation or epigenetic changes, they're like books about growth and division that get stuck open, continuously instructing cells to divide faster and faster. The interplay between tumor suppressor genes and oncogenes determines whether a cell becomes cancerous. It's like a complex balance between the accelerator and brakes in a car. Cancer often arises when the accelerator genes get stuck in the on position while the brake genes fail to function properly. Modern cancer treatment increasingly relies on understanding each tumor's specific genetic and epigenetic profile. Two patients with lung cancer might receive completely different treatments based on which particular books in their genetic libraries are damaged or incorrectly bookmarked. This personalized approach has led to remarkable successes, but it has also revealed just how many different ways cancer can arise and evolve. This genetic complexity sets the stage for our newest ally in the fight against cancer. Can AI outsmart cancer before it mutates again? Imagine having a chess grandmaster who could analyze not just the current board position, but simultaneously consider millions of possible future moves across thousands of different games. This is the promise that artificial intelligence brings to cancer treatment. The ability to process vast amounts of information and identify patterns that would take human researchers lifetimes to discover. Cancer generates enormous amounts of data. Each tumor contains thousands of genetic variations. Every patient has a unique medical history, genetic background, and treatment response profile. Traditional medical research, brilliant as it has been, resembles trying to solve a jigsaw puzzle with millions of pieces using only human eyes and hands. AI offers us the possibility of a super-intelligent partner who never gets tired, never overlooks details, and can hold countless pieces simultaneously while searching for connections. Machine learning algorithms are already helping pathologists identify cancer cells in tissue samples with greater accuracy than human examination alone. These AI systems have been trained on millions of microscopic images, learning to recognize subtle patterns that might indicate the presence of cancer cells or predict how aggressive a particular tumor might be. It's like having an assistant with perfect memory who has studied every cancer case in medical history and can instantly recall similar patterns. Drug discovery represents another frontier where AI shows tremendous promise. Developing new cancer drugs traditionally takes 15 to 20 years and costs billions of dollars. Much of this time is spent testing countless chemical compounds to see which ones might have anti-cancer properties. AI can simulate these experiments digitally, predicting which compounds are most likely to be effective before expensive laboratory testing begins. It's like having a crystal ball that can show us which keys are most likely to fit the locks we're trying to open. Perhaps most exciting is AI's potential to predict cancer evolution. Since cancer cells constantly mutate and develop resistance to treatments, scientists are using AI to model how tumors might evolve in response to different therapeutic approaches. These systems can analyze a tumor's current genetic profile and predict which mutations are most likely to arise during treatment. It's like playing chess against an opponent while simultaneously calculating their most probable next dozen moves. AI is also revolutionizing the analysis of medical imaging. Radiologists examining CT scans, X-rays, and MRIs are being assisted by AI systems that can detect subtle changes that might indicate cancer recurrence or treatment response. These systems don't replace human expertise but augment it, like giving a skilled detective a magnifying glass that can reveal fingerprints invisible to the naked eye. The challenge lies in the quality and quantity of data available to train these AI systems. Cancer affects people differently across various populations, but most medical databases have historically been skewed toward certain demographic groups. An AI system trained primarily on data from one population might not perform as well when applied to patients from different genetic or ethnic backgrounds. It's like training a translator using books written in only one dialect, and then expecting perfect translations across all variations of a language. Privacy and data sharing represent another significant hurdle. The most powerful AI systems require access to vast amounts of patient information from many different hospitals and research centers. However, protecting patient privacy while enabling data sharing for research purposes requires complex technical and legal frameworks that are still being developed. There's also the question of AI bias and decision-making transparency. While AI systems can identify patterns that humans miss, they can also perpetuate or amplify existing biases present in their training data. If historical treatment decisions reflected unconscious biases about age, gender, or ethnicity, AI systems might learn and replicate these biases. Additionally, many AI systems work as black boxes, making accurate predictions without clearly explaining how they reached their conclusions. In cancer treatment, where life and death decisions must be made, doctors and patients need to understand not just what the AI recommends, but why. The most promising applications might come from combining AI's pattern recognition abilities with human insight and intuition. AI can process vast amounts of information and identify potential connections, but human researchers and clinicians bring creativity, ethical reasoning, and the ability to ask unexpected questions. Together, they might achieve what neither could accomplish alone. As we develop these powerful tools, we're also confronting fundamental questions about the nature of cancer treatment itself, why a one-size-fits-all cure doesn't exist. Picture walking into a vast botanical garden, where every plant requires different soil, water, sunlight, and nutrients to thrive. Some flourish in sandy desert conditions, others need rich, moist earth. Some require full sunlight, others prefer shade. Now imagine trying to find a single treatment that would make every plant in the garden healthy. The task seems impossible, because each plant has evolved different strategies for survival. Cancer presents us with a similar challenge. What we call cancer is actually hundreds of different diseases that happen to share certain characteristics: the ability to grow uncontrollably and spread throughout the body. But each type of cancer has evolved different molecular mechanisms, different vulnerabilities, and different ways of interacting with the body's natural defenses. Breast cancer alone encompasses at least 4 major molecular subtypes, each requiring different treatment approaches. Hormone receptor-positive breast cancers depend on estrogen or progesterone to grow, so treatments that block these hormones can be highly effective. HER2-positive breast cancers overexpress a particular growth protein, making them vulnerable to targeted therapies like trastuzumab. Triple-negative breast cancers lack these common targets, requiring entirely different treatment strategies. It's as if we discovered that what we thought was a single type of plant was actually 4 completely different species that merely looked similar from a distance. The situation becomes even more complex when we consider that individual tumors are not uniform masses of identical cancer cells. Within a single tumor, different regions might contain cancer cells with different genetic mutations, different growth rates, and different treatment sensitivities. It's like discovering that what appeared to be a single plant is actually a community of related but distinct organisms, each with slightly different needs and vulnerabilities. Patient genetics add another layer of complexity. The same cancer drug can be processed differently by different patients, based on their genetic makeup. Some people metabolize certain drugs quickly, requiring higher doses for effectiveness. Others process drugs slowly, making them more susceptible to side effects. These genetic variations in drug metabolism, known as pharmacogenomics, help explain why identical treatments can produce dramatically different results in different patients. Age influences how both cancer and treatments behave. Childhood cancers often arise from different mechanisms than adult cancers and frequently respond better to treatment. Perhaps because young bodies are more resilient and cancer cells haven't had as much time to develop sophisticated resistance mechanisms. Elderly patients might have cancers that grow more slowly, but may also be less able to tolerate aggressive treatments due to other health conditions. The body's overall health status, what doctors call performance status, significantly affects treatment outcomes. A person who is physically fit might tolerate treatments that would be too toxic for someone whose body is already weakened by other illnesses. It's like the difference between a well-maintained garden that can withstand various treatments and a stressed ecosystem, where any intervention might cause additional damage. Even the location of cancer within the body matters enormously. Brain tumors face the challenge of the blood-brain barrier, a protective membrane that prevents many drugs from reaching the brain. Pancreatic cancers are often surrounded by dense fibrous tissue that makes it difficult for treatments to penetrate the tumor. Bone cancers exist in an environment rich in calcium and growth factors that can influence how treatments work. The timing of diagnosis also creates fundamental differences in treatment possibilities. Early-stage cancers might be completely curable with surgery alone, while the same cancer type, if caught later, might require combinations of surgery, chemotherapy, radiation, and targeted therapies, with significantly different outcomes. Perhaps most importantly, each person's immune system represents a unique ecosystem that influences how cancer develops and how treatments work. Some immune systems remain vigilant against cancer throughout life, while others seem to become less effective with age or due to various environmental factors. Immunotherapy works by enhancing or redirecting these individual immune responses, which explains why results can vary so dramatically between patients. Cultural and socioeconomic factors also influence cancer outcomes in ways that purely medical approaches cannot address. Access to healthcare, nutritional status, stress levels, social support systems, and environmental exposures all contribute to cancer development and treatment success. A perfect treatment that's unaffordable or unavailable cannot help those who need it most. This recognition of cancer's diversity has led medicine toward increasingly personalized approaches, but it has also forced us to reconsider what cure means. Chronic, not cured. Living with cancer in the 21st century. Think about diabetes, high blood pressure, or asthma, conditions that were once considered death sentences, but are now managed as chronic diseases. People live full, productive lives while taking medications to control these conditions. This represents a fundamental shift in medical thinking, from expecting complete elimination of disease to achieving long-term management that allows for quality of life. Cancer medicine is undergoing a similar transformation. While we continue to seek cures, we're also learning to think of many cancers as chronic conditions that can be controlled rather than eliminated. This shift reflects both our growing understanding of cancer's complexity and the development of treatments that can keep cancer stable for years or even decades. Some leukemias exemplify this new reality. Chronic myeloid leukemia, once uniformly fatal, can now be controlled with targeted therapy drugs like imetinib. Many patients take a single pill daily and live normal lifespans with their cancer held in check. The cancer hasn't been eliminated. It's been transformed from a rapidly fatal disease into a manageable chronic condition. Advanced kidney cancer, melanoma, and lung cancer, have similarly been transformed by immunotherapy and targeted treatments. Patients who might have had months to live are now surviving for years, returning to work, traveling, and maintaining relationships, while managing their cancer like any other chronic condition. Their scans might show stable tumors that neither grow nor disappear, existing in a kind of medical detente. This shift toward chronic management requires different skills from both patients and healthcare providers. Instead of the intensive, short-term approach of curative treatment, chronic cancer care resembles the long-term relationship building required for conditions like diabetes. Patients become experts in managing side effects, recognizing signs of disease progression, and balancing treatment intensity with quality of life. The psychological adjustment to chronic cancer can be more challenging than adapting to acute treatment. During active treatment for potentially curable cancer, patients often describe feeling like they're fighting the disease with a clear goal of victory. Chronic cancer management requires a different mindset, more like learning to dance with an unwelcome partner than trying to defeat an enemy. Families and caregivers must also adapt to this new reality. The crisis mode of initial diagnosis and treatment gradually gives way to a new normal that includes ongoing medical appointments, medication management, and the persistent uncertainty that comes with living alongside cancer. Support systems must be sustainable for years rather than months. Healthcare systems are slowly adapting to provide better chronic cancer care. Traditional oncology focused heavily on acute treatment, chemotherapy cycles, radiation courses, and surgical procedures. Chronic cancer care requires more emphasis on symptom management, quality of life assessment, psychological support, and coordination between specialists. It's like shifting from emergency medicine to family practice in terms of the ongoing relationships and holistic care required. The economic implications of chronic cancer care are significant. While the intense costs of initial treatment are substantial but time-limited, chronic management creates ongoing expenses that can last for decades. Insurance systems and healthcare policies are still adapting to this reality, trying to balance coverage for expensive long-term treatments with overall healthcare costs. Workplace accommodations represent another area requiring adaptation. Employees with chronic cancer might need flexible schedules for ongoing treatment, modifications to job duties during periods of fatigue or side effects, or adjustments to physical work environments. The Americans with Disabilities Act and similar legislation in other countries provide some protection, but navigating these accommodations requires understanding from employers and colleagues. Social relationships can be complicated by chronic cancer status. Unlike acute illnesses that have clear beginning and endpoints, chronic cancer creates ongoing uncertainty about the future. Friends and family members might struggle to understand how someone can simultaneously be doing well and have cancer. Social events, travel planning, and major life decisions all become more complex when viewed through the lens of ongoing medical management. The concept of living with cancer has also changed how we think about treatment goals. Rather than pursuing the most aggressive possible treatment, regardless of side effects, chronic cancer management often involves carefully balancing treatment intensity with quality of life. A treatment that extends life by 6 months, but makes those months miserable, might be less desirable than a gentler approach that provides fewer months, but allows for meaningful activities and relationships. This evolution in cancer care connects deeply with the emotional and psychological aspects of the cancer experience. The psychological war, fear, hope, and perseverance. Imagine waking up every morning knowing that somewhere in your body, cells that were once part of you have become your adversary. This is the psychological reality that millions of people live with daily. Not just the physical burden of cancer, but the mental weight of carrying an enemy within. The initial diagnosis often creates a strange split in time. There's life before the diagnosis, when cancer was something that happened to other people, and life after, when every physical sensation becomes potentially meaningful. A headache might be brain metastases. Fatigue could indicate disease progression. The body, once trusted and familiar, becomes a source of constant vigilance and worry. Fear takes many forms in the cancer experience. There's the immediate fear of death, of course, but also more subtle fears that can be equally consuming. Fear of becoming a burden to loved ones. Fear of losing identity as disease and treatment change physical appearance and capabilities. Fear of financial ruin as medical bills accumulate. Fear of losing control over fundamental life decisions as medical needs begin dictating daily schedules. Yet alongside fear lives hope. and the relationship between these two emotions is complex and ever-changing. Hope might initially focus on cure, the belief that treatment will eliminate cancer completely and allow a return to normal life. As treatment progresses and realities become clearer, hope often evolves. It might shift toward hope for extended time with family, hope for minimal pain, hope for maintaining dignity and independence, or hope for contributing something meaningful despite illness. Medical hope itself becomes a complicated construct. Doctors must balance honesty about prognosis with the recognition that hope is essential for psychological well-being and might even influence physical outcomes. The placebo effect demonstrates that belief in treatment can produce measurable physiological changes. Patients who maintain hope often tolerate treatments better, participate more actively in their care, and report better quality of life even when objective measures of disease remain unchanged. Social relationships undergo profound changes during the cancer experience. Some relationships deepen as friends and family members rise to provide support, discovering reserves of strength and compassion they didn't know they possessed. Other relationships become strained or even dissolve as people struggle to know what to say or do in the face of serious illness. Well-meaning friends might offer advice, share stories of others' cancer experiences, or simply avoid contact because they don't know how to handle their own discomfort. The concept of fighting cancer, while psychologically appealing to many patients, can create its own burdens. If cancer is an enemy to be fought, then disease progression might feel like personal failure rather than the natural progression of a complex biological process. Some patients find empowerment in warrior metaphors, while others prefer thinking of treatment as collaboration with medical teams or as learning to live alongside uncertainty. Guilt represents another common psychological burden. Patients might blame themselves for lifestyle choices they believe contributed to cancer development, even when scientific evidence doesn't support such connections. Survivors sometimes experience guilt about their good fortune when others with similar diagnoses have worse outcomes. Family members might feel guilty about feeling relieved when a long illness finally ends, or about continuing to enjoy life while their loved one suffers. The loss of future plans can be as devastating as physical symptoms. Career goals, retirement dreams, travel plans, and family milestones all become uncertain. This anticipatory grief, mourning losses that haven't yet occurred, but seem likely, requires its own form of psychological adjustment. Some people respond by accelerating important activities, trying to accomplish as much as possible while still able. Others find meaning in letting go of external achievements and focusing on present moments and relationships. Meaning-making becomes crucial in the cancer experience. People often describe their diagnosis as a catalyst for examining what truly matters in life. Priorities that seemed important before diagnosis, career advancement, material acquisitions, social status, might suddenly seem trivial compared to time with loved ones, spiritual exploration, or simple daily pleasures. This process of finding meaning doesn't necessarily make cancer worthwhile, but it can provide psychological resources for coping with its challenges. The role of spirituality and religion varies widely among cancer patients, but questions about life's purpose, the existence of something beyond physical existence, and the meaning of suffering become more pressing when confronted with mortality. Some people find comfort in existing religious beliefs, while others begin exploring spiritual questions for the first time. Still others find meaning in secular philosophies or in contributing to medical research that might help future patients. Healthcare providers are increasingly recognizing that treating cancer requires addressing psychological and spiritual needs alongside physical symptoms. Psycho-oncology has emerged as a specialized field focusing on the emotional aspects of cancer care. Support groups, counseling services and chaplaincy programs are becoming standard components of comprehensive cancer centers. The digital age has created new forms of cancer community through online support groups, social media connections, and patient advocacy networks. These virtual communities can provide support and information that might not be available locally, but they can also create information overload or unhelpful comparisons between different patients' experiences. As we grapple with these profound human dimensions of cancer, we're also looking toward the future to understand what role cancer might play in human health moving forward. Will cancer ever be truly curable, or just manageable? Picture humanity's relationship with infectious diseases over the past century. Smallpox, once a global killer, has been completely eradicated from the earth through vaccination. Polio, which paralyzed thousands of children annually, has been eliminated from most of the world. Yet, Other infectious diseases, influenza, tuberculosis, malaria, remain ongoing challenges that we manage rather than eliminate, adapting our strategies as pathogens evolve new forms of resistance. Cancer might follow a similar trajectory, but its fundamental nature makes complete eradication far more challenging than infectious diseases. Unlike viruses or bacteria that invade from outside, cancer arises from our own cells, through processes that are intimately connected to the basic mechanisms of life itself: cell division, DNA repair, and tissue regeneration. The mathematics of cancer present both hope and sobering realities. Every time a cell divides, there's a small chance of genetic errors occurring. With trillions of cells in the human body and billions of cell divisions happening throughout our lifetime, some degree of genetic mutation is inevitable. Most of these mutations are harmless, or are corrected by cellular repair mechanisms, but occasionally they create cells with cancer potential. As long as humans live long enough for these accumulated mutations to manifest, cancer will likely remain part of the human experience. Yet remarkable progress continues on multiple fronts. Vaccines against cancer-causing viruses, like HPV vaccines, that prevent cervical cancer, demonstrate that some cancers can indeed be prevented entirely. Early detection technologies are becoming more sophisticated, potentially identifying cancers when they're still easily treatable. Genetic testing can identify people at high risk, allowing for preventive measures or intensive screening. The emerging field of liquid biopsies represents a particularly promising development. These blood tests can detect microscopic amounts of cancer DNA circulating in the bloodstream, potentially identifying cancer recurrence months or years before conventional scans would reveal it. Imagine having a blood test as routine as checking cholesterol levels that could detect cancer in its earliest, most treatable stages. Artificial intelligence and machine learning continue to accelerate drug discovery and treatment optimization. As these systems become more sophisticated and gain access to larger datasets, they might identify treatment combinations and approaches that human researchers would never consider. The possibility exists that AI could discover fundamental principles about cancer biology that have eluded human scientists, leading to breakthrough treatments we cannot currently imagine. Gene editing technologies like CRISPR offer the theoretical possibility of correcting cancer-causing genetic mutations before they lead to malignancy. While current applications focus on treating existing cancers, future generations might benefit from genetic modifications that make cancer far less likely to develop. This raises complex ethical questions about genetic enhancement and the definition of normal human variation. Immunotherapy continues to evolve in sophisticated directions. Scientists are engineering immune cells in laboratories, training them to recognize specific cancer targets, and then infusing these enhanced cells back into patients. Coatal cell therapy has already shown remarkable results in certain blood cancers, and similar approaches are being developed for solid tumors. The possibility exists that future immunotherapies could create lasting immune memory against cancer. Similar to how vaccines protect against infectious diseases, prevention strategies are becoming more personalized and precise. Rather than general recommendations about diet and exercise, future prevention might involve detailed genetic analysis, microbiome assessment, and environmental exposure monitoring to create individualized risk reduction plans. People might receive personalized recommendations about nutrition, exercise, environmental exposures, and medical screening based on their unique biological profile. The concept of cancer dormancy offers another intriguing possibility. Scientists have discovered that many people carry microscopic cancers that never progress to clinical disease. Understanding what keeps these dormant cancers in check might lead to treatments that could induce permanent dormancy in more aggressive cancers, effectively transforming them from active diseases into harmless cellular passengers. Nanotechnology presents possibilities that sound like science fiction, but are becoming scientific reality. Microscopic robots designed to patrol the bloodstream, identifying and destroying cancer cells before they can establish tumors. Drug delivery systems that can target cancer cells with precision while leaving healthy tissue completely unharmed. Sensors that can detect single cancer cells and alert medical systems to their presence. However, cancer's fundamental connection to the aging process suggests that complete elimination might be neither possible nor desirable. The same cellular mechanisms that allow cancer to develop, genetic variation, cellular adaptation, resistance to programmed cell death, are also essential for normal development, wound healing, and immune function. Completely eliminating these processes might prevent cancer, but could also interfere with the body's ability to repair itself and adapt to changing conditions. The future of cancer care will likely involve increasingly sophisticated prevention, earlier detection, more precise treatments, and better management of cancer as a chronic condition. Rather than a single cure for cancer, we might see hundreds of targeted approaches for specific cancer types, genetic profiles, and individual circumstances. Quality of life considerations will become increasingly important as treatments become more effective at extending survival. Future cancer care might focus as much on maintaining normal life activities during treatment as on eliminating cancer cells. Treatments might be designed not just for maximum effectiveness, but for compatibility with work, family life, travel, and other activities that make life meaningful. The economic model of cancer care will need to evolve as well. Current systems designed around acute treatment episodes will need to adapt to providing decades of chronic management, prevention, and early detection, while requiring upfront investment, might prove more cost-effective than treating advanced disease. Global health equity will become increasingly important as sophisticated cancer treatments develop, ensuring that advances benefit all populations, not just those in wealthy countries with advanced healthcare systems. Perhaps most importantly, the future of cancer care will require continued evolution in how we think about the relationship between disease and human life. Rather than viewing cancer as a completely foreign invader to be eliminated at all costs, we might develop more nuanced approaches that recognize cancer as an opposite consequence of the same biological processes that make complex life possible. The journey through cancer, whether as patients, families, researchers, or healthcare providers, teaches us profound lessons about resilience, the preciousness of time, the importance of human connection, and the remarkable capacity of both science and the human spirit to adapt to seemingly impossible challenges. As we drift towards sleep tonight, we can take comfort in knowing that thousands of researchers around the world are working to understand cancer's mystery, that millions of people are living full lives despite cancer diagnoses, and that each small advance in understanding brings us closer to a future where cancer's power over human life continues to diminish. The story of cancer is ultimately a story about life itself, its incredible complexity, its remarkable resilience, and its precious fragility. In learning to live with cancer, we learn something essential about what it means to be human in a world where uncertainty is the only certainty, where hope and fear dance together, and where the greatest victories often come not from conquering our challenges completely, but from learning to carry them with grace, wisdom, and an abiding appreciation for each day we're given. The weeds in the garden of human health may never be completely eliminated, but with each passing season we become better gardeners, more skilled at tending what flourishes, more wise about what needs protection, and more grateful for the simple miracle of growth, renewal, and the enduring beauty that emerges, even in the presence of our greatest challenges. And in that recognition, perhaps we find not just scientific understanding, but a deeper wisdom about living fully in the face of uncertainty, finding meaning in the midst of difficulty, and discovering that sometimes our greatest strength comes not from eliminating our struggles, but from learning to transform them into sources of compassion, connection, and hope for others walking similar paths.